Everyone, and welcome to our first biology daily booster. What we're going to do in these daily boosters is just take a little section of the biology course so you can have a little look through, do a quick recap, and then at the end there'll be a little link over to where you can do a little quiz on a Google Classroom. Today we're going to have a look at microscopes. So the first type of microscope we need to know about is the light microscope. So do make sure you are familiar with the little diagram and you can label those parts, because this is one of the assessed practical tasks they can ask us about too. The key thing about microscopes is we're going to use them to observe structures that are too small to see with the naked eye. And the way the light microscope works is light passes through that specimen that we've placed on the slide on our stage there. Now, what we need to know are two calculations as far as microscopes go. The first one is total magnification, which quite simply is multiplying the eyepiece lens by the objective lens. So the question will say that the eyepiece has a magnification of times 10, the objective lens has a magnification of times 4. What is the total magnification? So 4 times 10 times 40. We need to know the steps of using a light microscope. Because this is one of the assessed practicals, they can ask you about the procedure, the names of parts, where you put things, for example. So we start off with our objective lens with the lowest magnification. You put your slide onto the stage, and then in order to focus, we start with the coarse focus knob, and then when you're pretty much there, use the fine focus just to fine tweak it, just to make it really clear. What we then do is we will rotate round the objective lens to the next magnification and then all the way to the highest magnification. You might need to focus it ever so slightly just at each of those stages. It is worth remembering that we always move the stage away from the objective lens as we do this. The second calculation that I mentioned earlier is all about using this formula here. So magnification is the image size divided by the actual size. They could ask you to calculate any of those three parts from information they give you in a question. So if it's the magnification, quite simply image size divided by actual size. Just make sure they are in the same units. If, however, they're asking you to calculate the image size, then we would have to rearrange. So use your triangle. So if we're working out the image size, that's the magnification times the actual size. Do make sure you use a calculator and do show your working in each case. The next key thing we need to know about are stains. What we find is that most cells and components of cells are actually colourless. So when we try to view them, you wouldn't really be able to make out anything because of that colourless nature. So instead, we apply these things called stains to make it easier to observe them. Now, some stains colour the whole cell, others highlight specific structures within the cell. And there are three stains you should really know the names of and what we use them for. So methylene blue, that's the nucleus in animal cells. Iodine, the nucleus in plant cells. And crystal violet we use for bacterial cell walls. Again, this comes under the practical side of things. So in order to apply the stain, you've got to put the cells on the slide, add the drop of the stain, and then we place the cover slip on top. Do know the name cover slip. If they ask you the name of the small piece of glass placed over a specimen on a slide, cover slip is what they are looking for. The next part we need to understand is the comparison between the two types of microscope that we look at, light and electron. And the key thing we're really looking at here is going to be resolution. So resolution, quite simply, is the smallest distance between 2 points where we can still see them as separate points. Anything smaller than 0.2 micrometers, we can't see with a light microscope. So that means we need something that can see much smaller structures and cells. And we used the electron microscope for this. The reason we use our electron microscope is it allows us to see more detail. We can reveal greater information on those subcellular structures. And we can understand the structure of viruses, because viruses are tiny little things. So using electron microscope lets us see what a virus looks like, and therefore we can develop drugs to actually combat the virus. As far as the electron microscope goes, there are two types: transmission electron microscope, or TEM, and the scanning electron microscope, or SEM. Key difference: the TEM has the most magnified images. The SEM, however, lets us see three-dimensional images of the surface. So we use them in different settings. If you want to see what the surface looks like in 3 dimensions, scanning electron microscope. If you want to see something in a lot of detail, so you need to zoom in an awful lot, magnify it a lot, we use the transmission electron microscope. One of the questions they could ask you is about a comparison between light and electron microscopes. So as far as the light microscope goes, then it's cheap and it's easy to operate. It's small, it's portable, so you can take it out into the field with you, but the resolution only goes to 0.2 micrometers. We can view specimens which are either living or dead, and it's very simple to prepare our sample. In the terms of the electron microscope, these are much more expensive than the light microscope. They're also very large. You can't take the electron microscope out into the middle of the rainforest, for example. But the positive, it does have a much greater resolution. You can see there 0.1 nanometers. Specimens do have to be dead though, so that's a downside. And it's quite complicated to prepare the samples there. So just bear in mind those kind of key points about them. So light microscopes, advantages: they're cheap, they're portable, but the resolution is lower than that of the electron microscope. So just make sure you have those options available that you can then discuss to compare those two microscopes properly. So the final thing for today then is to test your knowledge. So there's a little form over on Google Forms. I'll also paste that link in the description box, so you can just click on the link, go and see how you know this topic from today, and then I will see you again tomorrow for our next Daily Booster.